Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Today I'm going to present about the protein extraction and identification by gel electrophoresis and mass spectrometry from edible bird nest samples. So my name is Muhammad Khairul Afiq bin Ibrahim. My team member are Khairul Harisin and Muhammad Taufiq. So the purpose of this study is firstly to study and compare the protein profiles of different edible bird nests. Next, to investigate the EBN composition and its pharmacological benefits. Lastly, to study the protein extraction of different methods and its result. So now let's move on to sample type. The EBN from white nest swiftlets were harvested from the local birdhouse located in Batu Pahat and Kota Tinggi. They were 5 pieces of sample from each location ground into powder using a mortar and pestle, and only the powder sample without feather and dirt was cooked for protein extraction. So as for the extraction, the methods used are ultrasonic assisted protein extraction by trace HCl buffer, and Bradford assay for protein content. As for the ultrasonic assisted protein extraction, the EBN sample dissolved in deionized water, then the solution is filled into a tube in a beaker, then filled with ice. Then the solution will sonicated for 30 minutes. The EBN solution centrifuged for 10 minutes after that. Lastly, the protein was frozen and freeze-dried. As for the protein extraction by trace HCl buffer, the EBN sample were dissolved and extracted by 50 ml trace HCl buffer at 20 micromolar containing 25 micromolar EDTA at pH 7.5. Then the solution be filtered and centrifuged for 10 minutes. Lastly, the sample will be freeze-dried. Now let's move on to Bradford assay for protein content. The crude protein content extract from EBN using Bradford assay. Then the EBN protein pellet dissolved in 40 micromolar trace HCl at pH 7.5 buffer. Next, the 30 microliter of standard BSA solution and protein extract were added into 970 microliter of Bradford reagent. After solution well mixed, it is incubated for 5 minutes at room temperature. Lastly, the crude protein content was determined by triplicate assay. Now let's move on to the conclusion of this finding. So the finding of the study found out that the total protein content of EBN sample from Batu Pahat was significantly higher than the from Kota Tinggi for all extraction methods except for trace HCl bioanalysis. Throughout the method used, the water extraction produced the protein content at the highest, followed by the detergent lysis and bioanalysis. The finding indicated that EBN has abundance of water-soluble proteins. I think that's all from me. Thank you.
Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Taufiq bin Ahmad. Today, I will present about the analysis on the separation protein in edible bird nests. So, first of all, we have Bradford assay for protein content 1D SDS page in solution trypsin digestion, in gel trypsin digestion, liquid chromatography with tandem mass spectrometry, and lastly, protein search. For the Bradford assay for protein content, first, the crude protein content of protein extract from the edible bird nest sample was estimated by using Bradford assay. This protein pellet will dissolve in a buffer solution, and then protein extract were added into Bradford reagent in the solution and then it will incubated for 5 minutes at room temperature. As a result, the intensity of blue color from the protein complex formation was measured by using a UV-Vis at 595 nanometer. For the one-dimensional SDS page, first, the protein extract was mixed with SDS sample buffer. Bromophenol blue were added into the sample buffer and the mixture was topped up to 10 ml by distilled water. The gel was calibrated by a wide-range protein ladder from the molecular weight of 17 to 245 kilodalton. The voltage used for the protein separation is around 100 and 150 volt. And lastly, this process, SDS page, will undergo staining and de-staining process. For the in-solution trypsin digestion, we have 3 steps. First step is reduction, second step is alkylation, third step is digestion. So first of all, the protein extract react with dithiotryptol for disulfide bonded protein reduction. Then, protein extraction undergoes carbamidomethylation process by reacting with iodoacetamide. And then, trypsin solution was added into the protein solution for peptide bond hydrolysis at the carboxy group of lysine and arginine. And lastly, the peptide solution was acidified to pH 5 with formic acid and sent for LC-MS or MS analysis. For the in-gel trypsin digestion, first, the protein bands on the gel were cut into smaller pieces, and then the gel piece will wash with ammonium bicarbonate until the blue color of the gel was washed out. And then the small pieces of gel went through the reduction process by adding dithiotryptol, and then we add with iodoacetamide for alkylation, and then acetonitrile was added until the gel was shrunk and dried to complete dehydration. And lastly, the tubes containing the gel were sealed well and placed at an incubator around 37 degrees Celsius overnight. For the liquid chromatography with tandem mass spectrometry, the peptide solution was analyzed by a capillary liquid chromatography. A reverse phase with a flow rate of 10 microliter per minute was used for peptide separation. This system consists of 2 solvents. First, solvent A consists of 2% acetonitrile in water with 0.1% formic acid. Second solvent consists of 2% water in acetonitrile with 0.1% formic acid. And then the sample was analyzed in positive ion mode. Nitrogen gas and curtain gas were used for nebulizing. And lastly, the voltage of ion spray is 500 volt. The declustering potential is 50 volt, and the focusing potential is set at 250 volt. And lastly, for the protein search, the mass spectra of peptides were matched to the protein database from the National Center for biotechnology information using a basic local alignment search tool algorithm. That's all from me. Thank you.
I'm Kara Harisin. Will be presenting results from analysis of edible bird nest sample. So there are two methods involved. The first one is gel electrophoresis, which is shown in this figure, and the second one is mass spectrometry, which I will discuss later. So in this protein identification, there are two kind of methods used. The first one is in solution trypsin digestion, which use liquid chromatography tandem mass spectrometry. So this method uh, is obtained directly from the protein extract solution, while the second solution, while the second method, which is in gel trypsin digestion, comes from the gel segments. These gel segments, which are cut, processed, and further analyzed by mass spectrometry. So from the second method, we can link the type of protein with their exact position in the gel as shown in this figure. So from this gel electrophoresis, there are four methods used. The first one is ultrasonic. The second to fourth one is the detergent assisted. So from this figure, it can be seen that the detergent method is less intense compared to the ultrasonic. This may due to the detergent effect on the protein solubility. So the second one is the smearing effect in ultrasonic method, which it appears to be more smeared than these three detergent assisted methods. So this may due to the enzyme activity and also proteinous constituent exist. So from the in-solution trypsin digestion, there are many proteins identified. There are many proteins detected. Some of them are chitinase, which involves in influenza inhibition activity, and also COQ binding protein, which is responsible for body antioxidant production. While from the trypsin digestion, there are also many proteins identified. The trypsinogen, which is a pancreatic proenzyme in human intestine. As seen from this table, it is located at band number 10, which is at here. And then there are also three other kinds of proteins with their specific function. So from the mass spectrometry, from this example of mass spectra, the proteins are broken down into peptides, which gives the B and Y signal, which are actually the amino terminus and carboxyl terminus. So this B and Y signal is compared to the database for the identification process, such as these four kind of proteins. That's all. Thank you.